Welcome to the Deep Dive. Today we're tracking what really feels like a revolution happening right now in oncology. We're looking at how precision medicine and, well, artificial intelligence are fundamentally rewriting the playbook for treating prostate cancer.
That's absolutely right. If look across the sources we've got here, there is a clear, an undeniable shift
All
happening.
big shift.
Huge. We are moving decisively away from that sort of generalized chemotherapy towards these highly targeted strategies. The future, really, as you can see here, is all about combination approaches and driven by these amazing sort of guided delivery systems we call ADCS.
Antibody drug conjugates.
Exactly, ADCs.
So our mission today is to give you a clear roadmap of this progress. We're going to unpack the cutting-edge clinical trials, validate those successes with real-world outcomes, and then look forward a bit. Look at the incredible AI tools that are actually helping design the next generation of drugs. You're getting the essence of modern oncology delivered fast and thorough. Let's dive in. We should probably begin where the future is really being decided in the clinical trials, specifically focusing on these ADCS. We've called them guided missiles before, right? The antibody finds the cancer cell, and the specialized payload kills it locally. But the sources show these missiles are getting incredibly sophisticated.
Yeah, what's fascinating is the selectivity of the targets now. Our first subject here is this investigational drug, AZD0516. It's currently in an ongoing phase 1/2, a trial for metastatic castration-resistant prostate cancer, mCRPC.
Which is the really tough stuff.
Exactly, the really hard-to-treat cases where, standard hormone therapy has failed.
And AZD0516 is getting attention because, well, it's the first ADC to target STEAP2. Why STEAP2? Is it like a cleaner target than what we've seen before?
The data certainly suggests it is cleaner, yeah. STEAP, that stands for 6-transmembrane epithelial antigen of the prostate 2. Quite a mouthful.
Wow.
It's highly selective. We see it expressed on over 85% of prostate tumor cells across all stages. But here's the key thing. It's minimally present in healthy tissues outside the prostate.
Okay. So good therapeutic window.
Precisely. High confidence the delivery system will hit the right target and mostly leave healthy cells alone.
And the design itself sounds like, well, real molecular engineering. It combines the STEAP2 antibody, obviously, but the sources really emphasize the role of this specialized linker. It's not just connecting things, is it? It's a beta-glucuronidase cleavable linker.
That's absolutely vital. The linker is specifically designed only to release its toxic cargo, the payload, when it encounters the acidic, enzyme-rich environment inside the tumor cell.
So the poison stays locked down until it's delivered right to the target.
Exactly. Locked and loaded until it reaches the destination.
And that payload is exatecan.
Yeah.
For listeners maybe not deep in the molecular biology, what does exatecan actually do?
Right. So exatecan is a potent topoisomerase 1 inhibitor. You can think of topoisomerase as the enzyme that unwinds the cancer cell's DNA strand so it can copy itself and divide.
Yeah.
Exatecan basically stops that unwinding process cold, and the preclinical data shows this specific payload is like 3 to 10 times more effective at inducing cell death than related older drugs like Irinotecan.
That's impressive precision delivery.
Yeah.
But here's where the strategy gets, well, even more strategic. The combination approach. The trial's second module pairs AZD0516 with another drug, AZD9574. That's a brain-penetrant PAR-P1 inhibitor.
Yeah, and this is a classic example of seeking what we call synthetic lethality.
Wait, okay. Synthetic lethality, that sounds intense. How should we think about that combination in simple terms? What's the strategy?
Okay, imagine the cancer cell as a fortress, right? The exatecan payload from the ADC is like blowing holes in the outer walls. It's breaking the DNA.
Got it.
Right. Then the PAR-P1 inhibitor, AZD9574, comes in and blocks the construction crew. It stops the cancer cell from repairing those DNA breaks.
So you hit it, and then you stop it from fixing itself.
Exactly. By preventing the repair mechanism, you ensure maximum cell death. They work together, synergistically lethal, even though maybe neither one alone would be quite as effective.
It's a really complementary attack strategy. Very clever.
Mm-hmm.
And we're also seeing these combination strategies moving into the truly aggressive subtypes of prostate cancer too, right?
Right.
There's a phase 2 trial testing Sacituzumab, Tirumotecan, combined with Tagitanlimab.
Yes, and this specific combination is targeting patients with what's called aggressive variant and neuroendocrine prostate cancer, or NEPC.
Mm.
These are notoriously difficult tumor types because they often fail standard hormone treatments pretty quickly.
Leaving fewer options.
Precisely, forcing physicians often towards less targeted therapies.
So the combination here is a dual attack again. You've got Sacituzumab Tirumotecan, that's an ADC targeting TROP2, releasing another topoisomerase inhibitor. And then Tagitanlimab is the immunotherapy component, a PD-L1 inhibitor.
Right. And the rationale connecting them is really interesting. There's growing evidence suggesting that ADCs don't just kill cells directly. The cellular damage they cause can actually make the tumors more visible to the body's own immune system.
Oh, interesting. So the ADC sort of flags the target.
Kind of, yeah. It causes damage, creates signals, and then the PD-L1 inhibitor simultaneously removes the molecular brakes that usually keep the body's immune defenses suppressed. Let the immune system see the flagged target and attack.
Okay, that makes sense. And finally, pushing the envelope even further on this clinical frontier, we see bispecific ADCs, specifically TJ-Coil-1, currently in a phase 1 trial. Now, if standard ADCs are already so precise, why the need for this bispecific approach? What's the advantage?
Well, the main driver here is overcoming drug resistance and, tumor evolution. If a tumor manages to just stop expressing one specific protein target, it can basically become invisible to a standard monospecific ADC.
Right, it learns to hide.
Exactly. So TJ-101 hedges against that by targeting two different proteins simultaneously. It hits EGFR, which is known to drive tumor growth, and B7H3, which is an immune checkpoint protein that's, well, highly abundant in advanced prostate cancers, especially those that have become hormone resistant.
Targeting two independent pathways. That significantly reduces the tumor's chance of evading the treatment then. Plus, the sources highlight its delivery power. TJ-101 apparently carries 8 copies of the toxic payload.
Yeah, 8 copies.
And uses what's called a bystander effect.
Yeah.
Meaning it kills nearby cells too.
That's right. The released toxins can diffuse and kill neighboring cancer cells, even if those specific neighbors don't happen to express both or even either of the target proteins. It increases the killing zone.
That really is next-level multi-factor precision.
It really is. Now, okay, we need to transition a bit. We've talked about the bleeding edge, these novel trials. But we also need validation, right? Because novel targets are great. But the true measure of progress is often whether established treatments deliver on their promises outside of those tightly controlled research settings.
Absolutely. Real-world impact. Which brings us neatly to real-world data. Specifically, there's a study from Spain looking at Apalutamide for metastatic hormone-sensitive prostate cancer. This data seems to confirm its effectiveness in general practice.
And the results really do speak to the depth and, importantly, the speed of the response. 65% of patients achieved what are called ultra-low PSA levels. That's below 0.2 nanograms per milliliter within just 3 months of starting the treatment.
3 months? That's fast.
It is. And by 6 months, that number jumped to 71%.
So if 71% hit that goal in 6 months, does that level of speed itself stand out compared to, say, older treatments in the real world?
Absolutely it does. Speed of response is a key indicator of therapeutic effectiveness, especially outside the perfect conditions of a trial. But, what's arguably even more compelling here is the depth of that response. A remarkable 53% reached PSA levels below 0.02 nanograms per milliliter.
Okay, 0.02. That's significantly lower. Why is that threshold important?
It is significant. There's emerging research that strongly links achieving that ultra-low PSA level directly to exceptional long-term outcomes. We're talking longer survival, longer time without disease progression.
Wow.
So this real-world evidence gives immense confidence that the treatment really translates successfully into everyday clinical practice and delivers profound benefit.
And just as importantly, maybe, the quality of life metrics seem to follow suit. The data confirms that cancer-related symptoms actually improved over time. The proportion of patients reporting significant pain dropped pretty substantially, from 35% down to 22% over 6 months.
Yeah, and importantly, that happened while overall quality of life remains stable, so you're getting better cancer control without necessarily adding a huge burden from side effects.
That's the ideal combination.
It really is. And, this success isn't just isolated to Spain. The source also references a major U.S. comparative study involving nearly 4,000 1,000 patients. That showed apalutamide resulted in a 23% mortality reduction,
23%.
which translated to an 85.4% survival rate at 24 months. So the real-world validation seems robust and clear across different settings.
Okay, so we've covered the present and the immediate future of clinical treatment with ADCs and combinations, and seen powerful real-world validation for existing drugs like apalutamide. Now let's shift gears. How is the next wave of drugs going to be discovered? We're moving from analyzing trial data to maybe actually designing the therapy using AI.
Exactly. This brings us to PDGrapher. It's an artificial intelligence model developed over at Harvard Medical School. And this model represents really a fundamental paradigm shift away from the traditional approach to drug discovery.
Which is?
Well, the traditional idea, which often fails in complex diseases like cancer, is that you can successfully treat the illness by simply inhibiting a single protein target.
Right, the magic bullet idea, which doesn't always work for complex networks.
Precisely. The molecular networks in cancer are just too intricate, too redundant for a single hit to always be effective long-term. So PDGrapher tackles what's called the inverse problem.
Okay, inverse problem sounds technical. Break that down for us. How should we picture that difference?
Okay, think of it like this. Traditional drug discovery is maybe like tasting a million random cakes and trying to guess the recipe that makes the best one. It's screening vast libraries.
Right, lots of trial and error.
PDGrapher, tackling the inverse problem, sort of flips that. You input the molecular profile of a sick cell, the bad cake, if you will, and the AI predicts the exact set of ingredients or molecular targets that must be modulated, must be changed to bake a healthy cake.
Ah, so it predicts the strategy needed to restore healthy cellular function, rather than just finding one molecule that hits one target.
Exactly that. It's built as a graph neural network, which means it excels at mapping these incredibly complex relationships among genes, proteins, entire pathways. It understands the molecular architecture of the illness.
And the results seem pretty staggering in terms of efficiency. The benchmarks reveal PDGrapher ranks true therapeutic targets up to 35% higher than competing AI models.
Yeah.
And critically, it's apparently 25 times faster.
25 times faster. Yeah, and this rapid target identification isn't just theoretical. The model was validated. It correctly identified known successful therapeutic targets like KDR.
Which is VEGFR2.
Right, VEGFR2, a key target in angiogenesis, and also TOP2A, another topoisomerase involved in DNA replication. This confirms it understands genuine biology and points towards real clinical utility.
And this technological leap is being democratized, essentially. The software is open source.
That's huge.
Researchers anywhere can input their own cellular data and receive these complex target predictions.
Yeah.
That could really accelerate progress far beyond what any single lab could manage.
Absolutely. The ultimate goal here is using tools like PDGrapher to analyze patient-specific cellular profiles made from a biopsy, and then providing the blueprint for highly personalized combination treatments. We're talking about designing therapies specifically for the molecular architecture of an individual's illness.
Okay, so let's try and synthesize what we've deep dived into today. The clinical trials clearly show medicine is already pushing hard toward highly personalized, multi-targeted strategies, whether that's achieving synthetic lethality with something like AZD0516 or using bispecifics like TJ201 to overcome resistance.
Exactly. The clinical front is already embracing complexity and combinations. And then the preclinical world, powered by AI tools like PDGrapher, is already building the engine to design the next generation of those very combinations, perhaps even more effectively and personally tailored. The convergence of advanced targeted medicine and sophisticated technology is undeniable. It's really a remarkably rapid pace of progress we're seeing across oncology.
It really is. So here's maybe a final thought for you, our listener, to mull over. Considering how quickly drugs like apalutamide are being validated and showing profound impact in the real world, just how fast will these advanced AI tools like PDGrapher be able to translate their combination target predictions into successful, actual combination-based clinical trials? The timeline feels like it's only accelerating.
That really is the key question, isn't it? It will likely define the next decade of precision oncology. How fast can we bridge that gap from prediction to patient?
We'll be watching closely. Until our next deep dive, stay strong and fight on.